It takes future-focused vision to find ideas that become solutions to the world's most pressing concerns. Innovation and creativity are the sparks that ignite to transform lives and society, and these need space to grow.
We are home to these creative and innovative sparks. Our work goes beyond the disciplines, across our 9 faculties, 7 campuses, and being one of Africa's top business schools. As an African global university, we stand proudly among the world's top universities. We remain at the forefront of universal discoveries and provide authentically African solutions to the world's greatest challenges and opportunities. We are pioneers in our fields, from the humanities to cutting-edge technologies, continually seeking ways to improve the world around us. Our hybrid learning systems provide quality education that enables graduates to become adaptable game changers. We have molded nearly 500,000 alumni who have made their mark locally and internationally, and we continue to inspire and equip our students to uphold our firmly rooted culture of excellence. At the heart of what we do is our people. They are creative, critical thinkers who can shape society for the better, because that is how we teach, learn, innovate, impact, and live. We are the University of Pretoria.
Thanks for joining us online and here live in the audience for this 31st edition of the University of Pretoria Expert Lecture. My name is Barend Erasmus. I'm the Dean of the Faculty of Natural and Agricultural Sciences, and because our expert this evening, Professor Thulani Makhalanyane, is also in this faculty, I will also be your Program Director. Quick outline of the evening. I will shortly introduce Professor Tawana Kupe to do the formal welcome and introduce our speaker. Thereafter, we'll listen to Professor Makhalanyane, which is the real reason why we're here. And at the end, there will be opportunity for some short questions from the audience. Unfortunately, we can't do questions from our online audience in real time, but if you do have a question online, please submit it, and we'll capture it, get to Prof. Makhalanyane afterwards, and then make sure you get the answer. So our question session will only be for the live audience, and we'll have 2 roaming mics. Please don't use the microphone in front of you. Rather, put up your hand, and then I'll arrange to have a roaming mic join you. Afterwards, we'll have some light refreshments, and then if there's any questions that have popped up that you didn't have an opportunity to ask in the question session, Professor Makhalanyane is here to do that in person. Professor Tawana Kupe has been Vice-Chancellor and Principal of the University of Pretoria since 2019. He holds a D.Phil in Media Studies from the University of Oslo in Norway. In December 2019, he received an honorary doctorate from Michigan State University, and shortly thereafter, he also received a second honorary doctorate from the University of Montpellier in France. Professor Kupe has a notable publication record, having authored journal articles, books, and book chapters in his main disciplines. These disciplines are media studies and journalism. He's an active member of several civil society organizations and is a board member of a number of tertiary education networks, organizations, and initiatives around the world. It gives me great pleasure to introduce Professor Tawana Kupe for the formal welcome and introduce our speaker this evening. Please switch off your mobile phones.
Thank you, Professor Barend Erasmus, Dean of the Faculty of Natural and Agricultural Sciences and our Program Director tonight. Ladies and gentlemen, my role here is to explain the expert lecture series and then to introduce our expert, Professor Makhalanyane. So in very brief terms, the expert lecture series involves me having to do some work. It's a lot of work. It takes a lot of time. I have to choose 2 people every year to give an expert lecture on the subject of their expertise to enrich the university's intellectual life. So one of the speakers this year is Professor Makhalanyane. Don't ask me who the second speaker is. I have too many things to remember, but I've chosen the second person. So tonight you'll be the audience then for the expertise that Professor Makhalanyane can give you. I'm not allowed to go further than that. Now to an introduction of Prof. Makhalanyane. Thulani Makhalanyane is a full professor in the Department of Biochemistry, Genetics, and Microbiology, popularly known, I think, in us as BGM. Often sounds like a big German company. And DSI NRF Sachiche in marine microbiomics at the University of Pretoria, the place usually known as UP, which also stands for up. You know. He graduated from the University of the Western Cape, where he completed his postgraduate training, MSc cum laude 2009, and a PhD in 2013. Post-PhD, he moved to UP in 2013. In the ensuing period, his research has focused almost entirely on understanding the ecology of microbial communities in extreme environments. These habitats include soil microbial communities in the Antarctic dry valleys, the Namib Desert, and the human gut microbiome, with an increasing focus on South African geographically strategic regions such as the Southern Ocean. He has previously taught undergraduate modules in microbiology and genetics and supervises 20 postgraduate students at master's and doctoral levels. Prof. Makhalanyane has over 70 publications and over 2,500 citations at Index 27. He has co-authored papers in leading multidisciplinary journals, Nature, Science, Nature Microbiology, Science Advances, and others, and has presented keynote addresses at several international meetings, including the International Symposium on Microbial Ecology. He serves on the leading editorial boards within his discipline and is currently senior editor of the International Society for Microbial Ecology's ISME journal. It's a very high impact factor, and M Systems as well. In 2018, Prof. Makhalanyane was elected to the International Board of ISME and was appointed Director of ISME's Ambassador Program, which promotes outreach in over 100 countries. Since 2019, he served as a member of the NRF Basic and Applied Microbiology Rating Specialist Committee and was appointed convener in 2022. He also serves on several national and international panels representing South Africa in bilateral discussions. Prof. Makhalanyane has received several national and international awards, including the ISME Ambassador of the Year Award in August 2018. I think it's last year when you received an award at the NRF and we were drinking a lot of wine in Cape Town. Yeah, that is. I remember that. He's a previous Fulbright scholar, was awarded the prestigious T.W. Kambule NSTF Award, Emerging Researcher in 2015, the UP Exceptional Achievers Award in 2019 and 2020 to 2024, and the National Research Foundation Prestigious Award P rating in 2019. Ladies and gentlemen, Professor Makhalanyane.
Thanks, Professor Kupe, for the very nice introduction. And thank you very much for giving me this opportunity. This opportunity is really important to me because I've got my family here, and after tonight, they might finally know what I actually do for a living. So, and you don't know, my dad is probably very upset because he should be watching Scheme Sam or something like that. But I'm glad he came and is here. So I'll start first by acknowledging the people that have made this possible. There's a long list. I've got collaborators on campus. I've got lots of students that actually do the work. and I'm indebted to lots of funders that fund the research that we do. What we do, and you'll find out, is quite expensive. So having people that support the work that we do, which extends beyond the research, but includes also supporting the students that do the work, is really important. So let me take you through an outline of what we'll be talking about today. I'll start first by talking about biodiversity and the centrality of microbiomes. I'll then give an overview of the thematic research areas within our research group, Microbiome at UP. And then I'll focus quite a lot on the gut microbiome. And then after that, I'll move towards giving you some information on what we know about the state of African microbiomes. And then I'll talk a little bit about the research that we've started linked to the South African Gut Microbiome Project, and then move towards telling you all why we should be supporting a move to expand what we know about African microbiomes. And I'll propose essentially a roadmap of all the things that we need to do in order to expand microbiome research in Africa. When you see this picture, it means it's time for cocktails, and we're at the end. So when we hear biodiversity, this is typically what people think. We think legs, leaves, and wings. But this is not the full extent of biodiversity. And I hope by the time you leave here, you've got a better appreciation of what biodiversity entails. This image I got from National Geographic page, and this shows essentially the tree of life. If you look at this tree of life, it describes essentially how we diverged from some of our nearest ancestors around 700 million years ago. It tells you information regarding the number of species. For instance, if you look at vertebrates, mammals, and humans, there's over 60,000 species. Arthropods are well over 1.5 million species. Of course, as humans, we are doing our best to bring these numbers down quite actively. My dad was a history teacher, so I'll start with a bit of a history lesson. So if we take the Earth's 4.5 billion years and we compress it first in a year and then compress it essentially down in a 24-hour cycle, the first humans, you people, appeared in the last 40 seconds. Life first appeared in February, to be exact, and I don't know how we know this, on the 25th of February. Multicellular organisms appeared in August. But what you can see is that microbes, which are the first evidence of life, were first to appear, and without photosynthesis by microorganisms, we would not essentially have all the biodiversity that we have now. So hopefully this little history lessons makes it very clear that without microbes, we're essentially nothing. And from the very start of life to now, microbes control all facets of life. So this is what I want you to think when you hear the word biodiversity. This is the phylogenetic tree of life. A lot of the images here, of course, you can't see, but it essentially shows you the vast diversity of cellular life that's out there. The image I showed you first, which shows a human-centric tree of life, is only that small portion that's displayed over there. The vast majority of everything that's out there is microbial. Microbes are pretty important for everything, right? They're important in terms of controlling human health. They're important in terms of cycling biogeochemical cycles, carbon sequestration, nitrogen fixation. All of these processes are essentially driven by microorganisms. Microbes are also important because we all know about climate change, but microbes may potentially act as feedback mechanisms, which may further exacerbate the effects of climatic change. So this is what I actually want you to think about when you think about biodiversity and microbes. This image shows how we are all linked to the environments around us. Microbes link soil, plants, animals, and essentially health, right? From the soil that's below our feet, we essentially ingest and take in different types of microorganisms. There's a deep connectivity between agriculture and different ecosystems. That's why there's a prevailing movement to try and promote what is known as the One Health concept, right? And this concept at its core lays at the basis that we're all interconnected. And what happens to one aspect of us, whether it's in animals, has deep and profound effects on all of us. Some of you might know this image. These are the United Nations Sustainable Development Goals. These goals, the 17 of them, essentially provide a basis of all of the priority areas that we ought to meet if we are planning to move towards a world that's more just, free of poverty, and where we have more or less an equivalence in terms of how everyone is treated in society. I can tell you that each and every one of these sustainable development goals, you point to one, I can tell you where microbes are applicable. You talk about no poverty. The consumption of microbe-based products is one clear way towards solving food challenges. You think about the design decent work and economic growth. Biotechnology startups are an important way where we could further accelerate the development of our economy. And this relies on us solving fundamental challenges in terms of what we know regarding microbiomes. You talk about gender equality, biotechnology and microbiology literacy is at the core of a transformed and equivalent workforce where everyone has an equal basis towards essentially finding their own way through the world, where they are driven in terms of what they can achieve. So all of them, I'm not going to go through each and every one of these. Essentially, as if you look at this quote, you can see where microbes fit into each of the 17 SDG goals. So I hope with this, you guys are convinced that microbes are important. But if not, I'll keep going, and hopefully by the time I'm done, you'll be further convinced. So this is our research group, Microbiome at UP. And this is our thematic areas of research. So we've got a very wide portfolio of research interest and things that we focus on. We've split these research areas into 3 thematic areas. The first focuses on environmental and engineered systems. The second focuses on host-associated microbiomes, and I'll spend a fair bit of time talking about this. And the third talks to bioinformatics support and services. In the first thematic area, there's a wide portfolio of research that focuses on marine biodiversity. The SACHI chair that I lead looks at questions related to ocean acidification, carbon export, and carbon responses to production, and microbial responses to production. We have a wide portfolio of research where we essentially focusing on terrestrial environments. This image that's shown here shows how we have a very wide aridity gradient in South Africa, moving from areas that are hyper-arid to more mesic environments. Of course, we use these systems to answer a wide range of questions. One of the questions we're interested in, and we're using this aridity gradient as a proxy, is to understand with further desification what impact this will have on microbially derived ecosystem functions and productivity. We've got a wide research niche where we also focus on aquatic microbiomes. At the top there is an image from one of the recent papers that we published. In this paper, we looked at 2 different water systems that receive water from pure water that's impacted by agricultural pollutants, and also water that's impacted by pollutants that are human pollutants. We used microbes, and specifically viruses, to try and see if we can develop new molecular markers to determine the extent of contamination within these environments. So there's a very wide portfolio of research, and I won't go through all of them. In the second thematic area, we focus quite a bit on human microbiomes. And in the next few slides, I'll try and motivate to you why this is important. So I don't know if you guys have heard of human gut microbiomes, but it's evidently quite a popular area in terms of the popular press. These press clippings are just the research that I found in the last day that's been focused on gut microbiomes. You can see there's articles over there about how thousands of unknown viruses discovered in baby poo and why this is not necessarily a bad thing. There's a bunch of articles talking about gut bacteria in babies and how they can help us predict things like type 1 diabetes. So clearly, this shows that this is an emerging and very exciting area of research with lots of questions that remain essentially unknown. So what is leading to the growth in this research? So there's been, in the past 20 years, a lot of advances in the technologies of genomic sequencing. And these advances have been spurred by the Human Genome Project, which was around 2001. So what you can see that since the advent of modern sequencing approaches, we've had ready and easy access towards genomic sequencing, which was not available before. So the ability to sequence lots of different ecosystems is essentially helping us make more and more new discoveries. The second reason that's leading to this explosion in gut microbiome studies is the fact that the cost of sequencing have come down, right? This plot shows Moore's prediction, which was made in the 60s, that shows that the cost of sequencing is predicted to go down essentially. And we're now at a point where sequencing costs a little bit less than $1,000 per sample. It's still not free and it's still not cheap, but it's come down substantially from the $150+ million that it cost to sequence the first human genome. And what you can see now is that you can sequence a full human genome at less than $1,000. So there's also been improvements in our ability to analyze gut microbiome and microbiome data sets. We've had improved analytics and advances in techniques linked to artificial intelligence that have helped us to better understand the sequence data that are produced from all of these sequencing projects. So I'll focus on the gut microbiome. So why is gut microbiome research important? So the microbiome, first, is numerically quite abundant. And as you can see from this Nature article, microbes are described as our fellow travelers. So as you sit there, you are essentially surrounded by tons of microbes, whether you took a shower before you came here or not. I'll run you through a bit of facts. We know that, for instance, the genes in our microbiome outnumber those of our genomes 100 to 1. We've got about 3.3 million non-redundant genes in the gut, and that bacteria outnumber viruses 5 to 1, right? And these numbers don't necessarily indicate a state where people are unhealthy. These are just part of our natural gut flora. These are our natural microbiomes. You can see that things like the gut microbiome in terms of weight take up about 2.5 kilograms, right? That is just constituted by our microbiome. So if you are into weight loss, you can minus 2.5 kgs because that's essentially just the gut microbiome. So the microbiome and its metabolites interact quite profoundly with our immune system, with our neuroendocrine system, and also our digestive system. In this paper, which is quite a beautiful and eloquent paper, they showed that the gut microbiome targeted diets have the ability to modulate human immune status. The authors showed that diet intervention has the possibility of changing the status of the gut microbiome and have a direct effect on the diet microbiome immune axis. What they also show is that things like a high fiber diet has a possibility of changing essentially the function of the gut microbiome and improving the status of your immune system. The paper also shows that a diet that's rich in fermented food has the possibility of increasing the diversity of the microbiome and also decreasing other markers that may be linked to things like inflammation. So the microbiome and its metabolites is quite consequential. In that table that's highlighted, I've highlighted how one of the metabolites that are produced in combination with our bodies and the microbiome has a profound effect. So what's shown there, and I'm hoping you can see it, is that short-chain fatty acids have the ability to regulate the CD4 T cells and essentially have the profound effect of mediating the interactions between our bodies and the immune status. So the second plot that's shown here, in this, I'm trying to illustrate how what happens in terms of how the gut microbiome functions is also a combination of how our gut microbiomes interacts with our bodies, right? There are certain functions that your body is not able to perform on its own, and we rely quite heavily on the immune or well-functioning of the microbiome to be able to digest certain metabolites. So there's also been a lot of studies that's shown that an unhealthy microbiome is implicated in a bunch of physical and mental disorders. This study, which was a very large study carried out to look at the factors, how environmental factors shape the gut microbiome in a Dutch population, took a population of over 2,700 families comprised of more than 8,000 participants, and showed that things like diet, anthropogenic and environmental factors have a profound effects of influencing the gut microbiome. So this plot that's shown here, which looks a little bit complicated, so I'll explain it a little bit. So what you can see on the x-axis essentially shows diseases. And what you can see on the y-axis shows different types of microorganisms. The blue shows a positive significant correlation. And when you look at this plot, you can see that things like cardiovascular and metabolic diseases, gastrointestinal disorders, mental and neurological disorders are significantly positively correlated to several different gut bacteria, right? This shows the profound effect of the impact of the gut microbiome. So I've told you that the gut microbiome also has potential in terms of being a source of microbially derived therapeutics. So this review article shows that in populations that have a very high reliance on fermented foods, and I show here examples for two that are widely used in South Africa. We eat a lot of amasi and a lot of maheu, and these foods tend to have a lot of fermented products that are linked to the gut microbiome, right? So these foods, when you look at them, have got potential to be used as gut microbiome-derived therapeutics that can be used, for instance, to treat people that have gastric ulceritis and other gut microbiome disturbances. This plot that shows here essentially proposes a way forward in terms of how we can essentially screen these gut-derived therapeutics in order to produce gut microbiome-derived products. And one of the points I want to make is that a lot of these studies that you see here have often been done in populations that do not necessarily have diets such as the ones that we have in Africa, and do not have the extent of diversity that we have in Africa. So by screening African populations, we've got a higher potential of potentially finding novel prebiotics, probiotics, and other antibiotics just by harnessing the power of our gut microbiomes. So I'll come to a message that I want to make quite clear. And that is to propose a case in terms of why we need to study African microbiomes specifically. So what you can see here is a plot in terms of the current knowledge that we have regarding African microbiomes. At the top, you can see that when you put in a Scopus search of just microbiome, you see well over 88,000 papers that are retrieved. When you use the same search tool, but you put Africa to restrict the research to African-derived microbiome papers, you see less than 500 papers. It's essentially 446 papers as of 2 days ago. The papers span a wide variety of fields. You see a lot of papers that are targeted in terms of medicine, biochemistry, genetics, and molecular biology, and they span over 30 research fields. The equivalent search, when you look at Africa and microbiome, only spans 11 research fields. What this plot highlights is the clear knowledge deficit that exists regarding the status of microbiomes on the African continent. So when you look at this plot, essentially this figure shows you the patents that have been granted and the countries of origin, right? This is just a general patent search. And what you can see is that, of course, most of it is from places like China and the US and Japan. And all of these countries have control and have produced a large number of research data that leads to novel patents, and that Africa is quite low in terms of the contribution in terms of novel patents. This plot here shows essentially the patents that have been generated from Africa, and a lot of these come from South Africa. Although the pace in terms of the novel patents that are produced in South Africa is still minute when compared to international patents. So this is the current status when you look at the genomic data that's produced globally. And I'm hopeful that you can see the clear gap that exists, right? When you compare the data that's retrieved from international sequence repositories, what you see is that a lot of the current data is produced in places like the US, in Europe, and places in Asia. The data that comes from Africa is quite low. So we are quite, in terms of the knowledge that's produced, we're quite on the back foot. And when you take a closer look, you can see that the current data sets, this is just when you look at data that's linked to human gut samples, that a lot of the data that currently exists would come from Tanzania. You might wonder why Tanzania has a lot of human gut samples that are in international databases. This is because Western populations have used Tanzania as a proxy for looking at diets that are essentially free from modern type of diets. So they are a trusted model system for comparing Western urbanized diets to those of rural populations. So that's why you see quite a lot of sequence data from Tanzania. But when you look around the African continent, I mean, the numbers of these samples is really low. So for this reason, we've called for the establishment and launched the South African Gut Microbiome Project. As part of this project, we hope to generate a comprehensive sequence database that's focused on gut microbiomes. We want to use this project to identify the factors that essentially shape the gut microbiome in South Africans and understand how things like genetics, lifestyle, diet, and environmental factors impact on the gut microbiome. We want to use this project to investigate the relationship between the gut microbiome and also health outcomes. As part of this project, we're also starting to establish a repository or a biobank that's focused on gut microbiomes that can essentially be used for future research. We're also interested to use the SA Gut project that we've launched as a platform for collaboration with other South African researchers and international researchers that are interested in gut microbiome research. So I'll give you one example of a project that we've done that's linked to the gut microbiome project. So this study started about 6 or 7 years ago and was funded by the Medical Research Council. In this project, we essentially wanted to compare the gut microbiomes of people in Pretoria, urban populations that eat a lot of processed diet, to those from rural locations. So we chose a population in the Venda district as our proxy for a less modern or less urbanized location. So from this, we essentially collected stool samples. And I could say, this 100 samples was probably the hardest 100 samples we ever had to collect, because for some reason, South Africans are very suspicious when you ask them for their stool samples. So what we did was we used a combination of approaches to study the organisms that are present in the gut microbiomes of these individuals, and also study the related functional processes. So I won't go through this in very great detail, but what these plots show is that when we looked at comparing the diversity of organisms that are present in the guts of urban and rural South Africans, we saw that the gut microbiomes of rural individuals was much higher. Higher diversity in the gut is often equated with higher stability. That means that your gut is functioning optimally. So the complicated plot that's shown here describes essentially how microbes within the gut interact with each other. So when you look at this plot, you can see that when you look at the rural networks, there's a lot of interactions between the gut microbiomes. Whereas when you look at the urban plot, there's less interactions. What this indicates is that the gut microbiomes in rural populations is a lot more synergistic. They interact with each other a lot more, and that again is a proxy for better health. And then we try to understand whether this difference in diversity within the gut bacteria that are present actually leads to functional differences. So there are a lot of different functions that we can derive from the gut. But what we looked at was assessing how antimicrobial resistance differs when we look at urban and rural individuals. And what you can see, this plot shows you essentially genomes that we retrieved from our data set. And you can see that there is a clear difference between rural, that's my nephew, between rural and urban individuals. And you see a lot of, there's a lot more bacteria that have capacity for antibiotic resistance in urban populations compared to people from rural locations. And when you look at linking the diversity of organisms that harbor antibiotic resistance genes, you can see that when you look at the putative resistance genes, there's a lot more resistance genes in the urban location compared to the rural individuals. So if you remember to the networks that I showed you previously, we saw that there's a lot more interaction in the rural individuals. So this further confirms that a high diversity ecosystem equates to essentially more stability. So we've started to prospect the data set that we retrieved in this study, and this work is quite ongoing. So we looked at gene editing mechanisms, and these are known as CRISPR-Cas systems, from uncultivated bacteria that are present within the guts of urban and rural individuals. I won't take you through all the details, but what we're seeing is that there's a high degree of novelty regarding gene editing systems that are present in the gut microbiomes of rural and urban individuals. And this plot essentially shows you a comparison when we look at whether they're novel or not, and compares urban individuals to data that's available in international databases. So what this shows is that there's a high degree of novelty, and high potential to explore the gut microbiomes of urban and rural individuals. So there are a lot of priority themes and a lot of areas that need to be studied when we look at African microbiomes. We would advocate quite strongly towards a push towards looking at the implications of the gut microbiome on health. There's a clear need to look at the implications of the gut microbiome on biodiversity. As I showed you at the start, microorganisms control essentially the status of human and animal health. There's a strong need to look at the effects of the microbiome on agriculture. We've got ongoing projects where we're looking essentially at whether different forms of agricultural production lead to microbiomes that have got more resistant bacteria, antimicrobial resistant bacteria, when compared to different classical forms of agriculture, for instance. And there's a strong need towards focusing on the gut microbiome, because if you're interested in things like conservation, you cannot really start to look at how to conserve different ecosystems if you do not look at microbiomes. Equally, you can look at the status of the microbiome and use it as a proxy towards determining whether a system is at a point of being disturbed or altered. Because of the metabolisms that microorganisms have, they respond quite rapidly, so they are a good proxy towards checking whether an ecosystem might be at a point of disturbance. So we've proposed a roadmap in terms of what we need to do to look at microbiome research in Africa. So we've started to set up coordinated research networks with colleagues in other African countries in order to essentially create Africa-centric microbiome research networks. The first of these activities will take place in around 2 weeks when we go to Cote d'Ivoire towards establishing the first research network that's focused on microbiomes. We feel that another thing that we need to do as Africans is be quite active in terms of proposing policy interventions and producing documents that serve to guide our respective governments on the importance of looking at microbiomes. So these policy documents, for instance, might focus on developing science plans. And these science plans might develop priority areas that are then the focus of microbiome research. Of course, I talked a little bit about the infrastructure that led to the development of microbiome research. But this infrastructure is often not available on our continent. And part of the roadmap that we see is to advocate in terms of generating research funding that would help the establishment of coordinated infrastructures, in a way, different network hubs, where sequencing would be able to take place throughout the African continent. Together, we see these 3 pillars as more or less a way or a roadmap in terms of decreasing that gap that I showed you in terms of the research outputs that are produced internationally versus what's produced in Africa. So just in summary, through this talk, I've tried to explain to you the importance of biodiversity and the central nature of microbiomes. I hope you're convinced that microbiomes are the most important thing in the world, and there's no research that ought to be done without looking at the effects of microbiomes. I hope I've convinced you of the need to strengthen efforts to focus on African microbiomes, because there is a clear need in terms of the available data and the diversity of our ecosystems that are unstudied could potentially hold biotechnological solutions that are as yet untapped. I'm inviting some of you that have tons of money, maybe under your couches, to give us and partner with us in terms of expanding microbiome research on our continent. And it's that time. This is where I need a stool.
Thank you very much, Professor Makhalanyane. I've had a few really profound and additional insights, even though I'm very familiar with what this group does. There's been quite a few little bits that have added that have got me thinking and making connections. I'm quite excited with what he's presented here, and the opportunities are certainly vast. Before we move to the question session, this is just where I want to highlight that this level of performance, this level of expertise, this level of insight doesn't happen by itself. And I'm not just talking about academic networks, and obviously that's the university environment, but also I'd like to extend a little bit prematurely before I get to the formal thank yous, but a big thank you to the family and the friends that support Professor Makhalanyane outside of the university to get this done. So thank you very much for that. Right, let's move into a question session. There's a roving mic, so if you have a question, there's a question over there, so the roving mic on the way there. And if there's a second question, maybe we can get the mic to that person in the meantime. Thank you very much. So there's the first question. Then we have the second question over here. First question.
Good evening, everyone. My name is Masehla Mailula. I'm an accounting science final year student. So I kind of have 3 questions in a sense. So my first question is, okay.
Pick your favorite question.
They're all my favorites, but what incentive would you say there is to invest in microbiome research to the layman such as myself in terms of how does it benefit me? Does it give me more energy? Does it make me more healthier? And what potential do you see from this research essentially moving forward? Thank you.
So this is essentially a whole different talk, right? But there is a reason why if you look at places like the Bay Area in California, you see large companies developing with investments of hundreds of millions of dollars focused on microbiome. The health implications of looking at microbiomes are quite profound. I talked a little bit in my talk in terms of how microbiomes are important for digesting certain metabolites, right? So things like precision medicine, you rely quite a lot in terms of your microbiome. If you don't have a good understanding of what's actually in there, it's hard to produce drugs that are tailor-made. And this is where the world is moving towards having personalized medicine, towards in terms of treating different ailments, right? So that's one area, why it would be important for anyone to focus on the microbiome. So for different insurers that are looking at things like how risky you are towards developing certain cancers, for instance, right? There's easily a place where when sequencing becomes easily available, people would submit their genomic sequence data, and we could use the same artificial intelligence and predictive models to see how at risk they are towards developing certain cancers, for instance. So there's a lot of applications.
Thank you so much. Good evening, everyone. My name is Mashirin Umalo, and I am an IP specialist from the Agriculture Research Council. Prof, in your presentation, you indicated a patent trend. So I just needed clarity on whether that search was a general one, or you did a microbiome-specific patent search.
It was a general one. Okay. Because, I mean, when I looked, I could not find specific patents, but I'm sure more specialized patent searchers would get a better idea. But this was a general one.
No, I agree with the trend. I just wanted clarity on that. Now, the second one on the roadmap with regards to policy interventions. Are you aware of any interventions that have been made in South Africa or in Africa? And are you also involved in those policy interventions? Thank you.
So it depends on the sector. The work that I talked about here was on gut microbiomes. But in another hat that I wear, with Gilbert Seeko over there, we helped develop a science plan for the South Atlantic that included at the base of that science plan why we need to focus on African microbiomes, right? So that's one example. And that science plan went on to inform the solicitation call documents for the EU-funded projects that we have now. So that's one instance of a policy intervention. But I think there's lots of potential towards developing ones that are much more focused, for instance, on health and other sectors where the microbiome has applications. Thank you.
Thank you very much. We've got one question, and then thereafter the second question in the row down here. Thank you.
Good day, everyone. My name is Kavel from Bioinformatics Honors. Thank you, Prof, for your presentation. I have a question about, you mentioned that there was an interaction between the bacteria taken from the rural and urban area. What is this interaction and how was it determined?
Okay, so the network plot that I showed essentially shows correlations among different bacteria. So you can correlate, essentially compare, for instance, if you look at yourself, you are 100% identical to yourself. If you compare yourself to someone else, you might have lower similarity, for instance. So it's a predictive model in terms of combining those interactions. The interactions that I showed here are, again, like it's a prediction, so we don't really know whether if we were able to isolate those microorganisms, whether those interactions would occur in real time in the same way that we predict that they would. Right? But the hurdle is that a lot of the microorganisms that I talk about and I've described, we know them at a sequence or molecular level, but we've not really isolated them in the lab. So we can't really do those types of mechanistic studies to study in real time those interactions.
Thank you very much, Barend. And Prof, thank you for exposing us to the importance of stool as we know it. And I think also your presentation is intriguing. So Barend, forgive us for having 5 questions, because in all honesty, we are being exposed to problems of modernity as well as we know them. My interest also, as we are delving into this exciting presentation you've just delivered, and by the way, okay, I'm Palsa Skajani from the Human Sciences Research. So I'm picking an interest really on the spatial development and demands as you've presented on rural versus urban spaces. And I think the urban spaces are really are fast becoming a highlight in terms of first food security or insecurities, but also now you're taking us further with the gut health or gut microbiome. So I'm quite interested also on the aspect of partnerships. So if we are to really invest in ensuring the success of this project, particularly from the policy interface and also understanding the project or the project in terms of the microbiome in Africa, what would be the kinds of partnerships and investments that would be necessary? And perhaps you might not have the answer to the policy interventions, as I've mentioned, rural versus urban space developments, because I'm also imagining that data that is coming rich is from East Africa, Tanzania, but West Africa also ingests a lot of fermented foods. So it's quite interesting what the insights for future would tell us. Thanks.
Yeah, I think you make a very good point. I mean, there's a lot of variability. That's why what we are advocating for first is having sort of proposals that are tailored to the specific country, because in terms of research priorities, what might be important for someone in Cote d'Ivoire might not necessarily be the same for someone in Pretoria, right? But by creating these networks, we can more or less provide a blueprint in terms of what we all need to do separately within our own spaces, but then also together, because there are certain things, for instance, which we can provide leadership on at a continental level, and that leadership might be in terms of how we interact with places like the African Union, for instance. We could, for instance, develop policy guidelines that essentially set the limit in terms of if you're having specific diets, how much of a certain food would be equivalent and enough to give you high fiber intake. And that that variable would be dependent on the prevalent diet within that location, right? But overall, in terms of the way the guidelines are phrased, they could be distinctive in each of the different regions where microbiome research is actually practiced. Hopefully that answers your question.
Thank you very much. I think time-wise, we've got 2 final questions, and any leftover, we will do so informally over light refreshment. So, Professor Makhalanyane.
Thank you, Chair, and thank you, Prof, for such a very insightful presentation. And my interest is mainly on the link between the environmental microbe and the gut microbe, because we know that the climate change impacts the plant and the soil microbes to a large extent negatively. So I just want to find out if you have done the correlation between the environmental microbes and the gut microbe, and also if the microbes can be trained, to say, you've been living in rural Venda for so many years, consuming this diet, and how far can that take you in terms of, sustaining your gut if you ever change your diet, and if the environmental factors, to what extent they do affect your gut microbe. Thank you.
Yeah, so we haven't looked at environmental factors directly in part of our study. In the example that I showed, we essentially asked people what type of diets they take in, and what you could see clearly is that people in cities take much more processed diet, whereas the people in the villages that we looked at would rely on more sustenance, and that diet included quite a lot of fiber, for instance, which might explain the differences in the variables, right? So we know that the gut microbiome is quite a dynamic system. There's been examples of studies where they've taken African American populations and the Hadza in Tanzania and essentially shifted their diets, right? And the African Americans have got a risk of contracting certain cancers, for instance, that are at a much higher proportion compared to Africans. So when they looked at what the effect of changing those diets are, is that those markers that are linked to the development of those cancers came down quite markedly in a very short amount of time, right? So hopefully that kind of shows the dynamic nature of the microbiome, and the fact that essentially if you eat a good diet, like I'm sure your mother told you, rich in fiber, rich in veggies, that has a way of promoting good gut balance and also reducing the possibility of certain cancers.
Thank you, Barend, and thank you, Thulani, for a nice talk. Good afternoon, everyone. I am Lucy Muleleki, professor in BGM. Thulani, I would like to get your view. No doubt we live in times of pandemic. We've just gone over COVID-19, and all predictions are that there will be another one around the corner. What, in your view, is the role of, or the importance of studying microbiomes in this time of pandemics?
Yeah, so, I mean, you make a very good point. And in this talk, of course, we know how one particular virus has essentially upended our lives, right? So if you look at the literature, people predict that the next great pandemic is the crisis of antimicrobial resistance, right? So with that, there's a clear need at looking at deriving much more novel antibiotics that are able to circumvent this crisis that we likely just about to go into. So that's one clear importance of studying the gut microbiome. But yeah, I mean, I could go on in terms of other applications, but in terms of antibiotic resistance, that's the next crisis, and that needs to be the focus of our target in terms of looking at gut microbiomes.
Thank you very much, Professor Makhalanyane. I think that brings us to the end of our question session. Anything else for our online audience, please make sure you get that in the chat line. We'll get back to you with an answer. I know there's one question there at the back, but let's leave that for light refreshments and in person. Friends, colleagues, the only thing that remains for me to do is to wrap up and close our proceedings here. Again, personal thank you to Professor Makhalanyane. It's easy to be a highly specialized scientist with high impact at a place like the University of Pretoria, and indeed we've got many such scientists. What we've seen tonight is taking that deep expert niche knowledge and putting it in the context of the existential and everyday challenges that we face as humanity. I think that systems perspective, that opportunity to influence a transition, a just transition to more sustainable futures, that is truly the mark of a master of his own craft. So Professor Makhalanyane, thank you very much for the insights and the expert opinions that you bring to this audience and elsewhere. Also, a heartfelt word, thank you, Professor Thulani Makhalanyane, for opening and welcoming Prof. Makhalanyane and giving us the context of Professor Makhalanyane's background and CV and how he's gotten to this particular point. Thank you very much for your time and your insight. An event like this doesn't happen by itself. Big thank you to Pure Cafe for catering. Big thank you to AJR for audiovisual matters, and iScape, thanks for the photographs. And Lynette Smith, thank you very much. I know you do many of these events, and you don't want to be thanked, but I'm purposefully doing it this evening. Thank you very much for initiating, arranging, and making sure it runs as smoothly as it does. Thank you very much. The only thing that remains for everyone in the audience, please join us for light refreshments outside. And those online, have a wonderful evening. And it's not quite weekend yet, but I hope you have a good weekend. All the best. Thank you.
It takes future-focused vision to find ideas that become solutions to the world's most pressing concerns. Innovation and creativity are the sparks that ignite to transform lives and society, and these need space to grow.
We are home to these creative and innovative sparks. Our work goes beyond the disciplines, across our 9 faculties, 7 campuses, and being one of Africa's top business schools. As an African global university, we stand proudly among the world's top universities. We remain at the forefront of universal discoveries and provide authentically African solutions to the world's greatest challenges and opportunities. We are pioneers in our fields, from the humanities to cutting-edge technologies, continually seeking ways to improve the world around us. Our hybrid learning systems provide quality education that enables graduates to become adaptable game changers. We have molded nearly 500,000 alumni who have made their mark locally and internationally, and we continue to inspire and equip our students to uphold our firmly rooted culture of excellence. At the heart of what we do is our people. They are creative, critical thinkers who can shape society for the better, because that is how we teach, learn, innovate, impact, and live. We are the University of Pretoria.